In 1889, paleontologist Othniel Charles Marsh described a large skull from the Lance Formation of Wyoming and gave it a name that has appeared on every dinosaur poster, in every natural history museum, and in every children's book about prehistoric life for the 135 years since. Triceratops horridus. Three-horned face. Horrible. The skull had two large brow horns above the eyes, a shorter nasal horn on the snout, and a solid bony frill extending back over the neck. 2 years later, Marsh described another ceratopsid skull, this one with a longer frill perforated by two large oval openings, and named it Torosaurus latus. For over a century, the two animals were treated as separate genera, both found in the same formations, living at the same time, representing two distinct branches of the ceratopsid family tree. In 2010, two paleontologists proposed that the second genus had never existed at all. Torosaurus, they argued, was simply Triceratops growing old. John Scannella and Jack Horner at Montana State University spent years examining ceratopsid skulls from the Hell Creek and Lance formations and noticed a pattern that had not been fully appreciated by earlier researchers. Triceratops skulls from younger individuals had thick, solid frills with no openings and horns that curved in one direction. Skulls from older individuals showed progressive thinning and remodeling of the frill bone, changes in horn orientation, and in some cases, early stages of what appeared to be fenestration, the bone becoming porous and beginning to open into gaps. The developmental trajectory they traced pointed directly toward the morphology that had been assigned to Torosaurus. And crucially, no juvenile Torosaurus specimens had ever been found anywhere. Torosaurus and Triceratops never appeared together in the same deposit at comparable life stages. The most parsimonious explanation, Scannella and Horner argued, was that the two genera were one animal at different ages. The frill did not remain static across a lifetime. It was a structure in continuous transformation. The reaction from the paleontological community was immediate and divided. If the hypothesis was correct, then Torosaurus, named in 1891, 2 years after Triceratops, would fall into synonymy under the earlier name, since taxonomic rules give priority to whichever name was published first. Nicholas Longrich and Daniel Field published a counter-analysis in 2012, examining the histological evidence in skulls from both genera. Bone histology, the microscopic internal structure of bone, reveals growth stage with considerable precision, because the pattern of growth rings and the density of vascularization change in predictable ways as an animal approaches skeletal maturity. Longrich and Field reported that some Torosaurus specimens showed histology consistent with an animal that had already reached full skeletal maturity, meaning the dramatic frill transformation would have had to occur after growth stopped, rather than during active growth. By their reading, the two genera were genuinely distinct animals that happened to live in the same place at the same time and looked somewhat similar because they were closely related. The debate has not been resolved to most researchers' satisfaction. What it forced, regardless of its outcome, was a much closer examination of how dramatically Triceratops itself changed across its lifespan. And that examination revealed layers of biological complexity the static museum mount had never suggested. Work by Horner and Mark Goodwin, published in 2006, had already begun documenting the scale of ontogenetic change in Triceratops skulls before the Torosaurus controversy erupted. Baby Triceratops specimens, identified by bone histology and by their small size, had proportions that differed radically from adults. The brow horns in juveniles were short and curved backward rather than forward over the face. The nasal horn was barely present. The frill was relatively small and had a different outline from the adult form. As the animal aged, the brow horns grew substantially, rotated forward, and in the oldest individuals began curving slightly downward again at the tips. The nasal horn changed shape multiple times across ontogeny. The frill expanded, thickened at its base, and developed epiossifications along its margin, small bony projections that varied between individuals in number and shape, but remained consistent within a single animal. An isolated specimen encountered at 3 different life stages could easily be assigned to 3 different species by a paleontologist without comparative material. The skull of Triceratops was not a fixed adult structure that juveniles were working toward. It was a dynamic system, continuously remodeling itself in response to biological signals that researchers are still working to understand. There is no Triceratops DNA. The animal lived between approximately 68 and 66 million years ago, right at the end of the Cretaceous period, and the genetic material is gone by the same enormous margin that makes T-Rex, Velociraptor, and every other Mesozoic dinosaur genetically unrecoverable. The Hell Creek Formation, where the great majority of Triceratops material has been found, spans parts of Montana, North Dakota, South Dakota, and Wyoming, and the preservation conditions are excellent for bone. But preservation of organic molecules at geological timescales is a different problem entirely. The theoretical upper limit for DNA survival under optimal cold and dry conditions is around 1 million years. Some studies suggest the real limit is lower. Triceratops died 68 million years before that ceiling, under conditions that were warm, seasonally wet, and biologically active. No nucleic acids survived. The genome of Triceratops, the complete biological instruction set that produced the horns, the frill, the 3-ton body, and whatever metabolism and behavior the animal possessed, is gone, and it is not recoverable. What is not entirely gone is the chemical record preserved inside dense, mineralized bone. Following Mary Schweitzer's 2005 discovery of apparent soft tissue structures inside a Tyrannosaurus rex femur, researchers began systematically testing other large dinosaur material for similar evidence of molecular preservation. Ceratopsid bone proved to be among the more productive sources. Schweitzer and colleagues reported finding what appeared to be original protein fragments, primarily collagen, preserved inside the cortical bone of ceratopsid specimens, and a 2013 study examined structures interpreted as dinosaur osteocytes, the cells embedded within bone matrix, from Triceratops and other ceratopsids. The study reported morphological and chemical signatures consistent with preserved original cellular material, including what the authors interpreted as remnants of protein chemistry. The findings attracted significant criticism. Some researchers argued the structures were microbial contamination, biofilms that had colonized the bone cavities long after the original tissue degraded, or diagenetic artifacts produced by chemical changes in the sediment over millions of years. The same arguments had been made against Schweitzer's T-Rex results, and were eventually addressed through a series of chemical and isotopic tests that made contamination increasingly difficult to sustain as an explanation. The debate over ceratopsid molecular preservation has not yet reached the same level of resolution, but the work has established that the dense, well-vascularized bone of a large ceratopsid is worth examining at the molecular level, and that if original proteins survive anywhere in the dinosaur fossil record, the bone of a Triceratops is among the reasonable places to look for them. The origin of Triceratops is a story that begins on a different continent and with a very different kind of animal. The ceratopsian lineage, the entire group of horned and frilled dinosaurs, evolved in Asia during the early Cretaceous from small, lightly built herbivores that show almost none of the features the group would later become famous for. Psittacosaurus, found in enormous abundance in early Cretaceous deposits across China and Mongolia, is among the earliest and most primitive ceratopsians known. It had no horns, no bony frill, and walked primarily on its hind legs. What it did have was a narrow, parrot-like beak for cropping tough vegetation, and the early outlines of the distinctive skull architecture that would eventually produce the ceratopsid face. From ancestors like Psittacosaurus, the ceratopsian lineage produced Protoceratops, the sheep-sized animal found alongside Velociraptor in the Mongolian Gobi Desert, and then, across the intermittent land bridge connecting Asia and North America through the shallow Bering region during much of the Cretaceous, the group crossed continents and diversified into the extraordinary variety of horned dinosaurs known from North American deposits. The North American ceratopsid radiation was one of the most elaborate evolutionary experiments in the history of large vertebrates. Within roughly 20 million years, the group produced an array of skull shapes and ornamental structures with no parallel in any living animal lineage. Styracosaurus had a frill ringed with long spikes radiating outward like a crown. Pachyrhinosaurus replaced the nasal horn with a large, rough-textured bony boss. Cosmoceratops bore 15 distinct horns and hornlets arranged across its skull and frill margin. Pentaceratops carried a frill so enormous that its skull constitutes the largest known skull of any land animal that ever lived. All of these animals evolved from the same Asian immigrant stock within a compressed geological interval, diversifying in response to ecological pressures and sexual selection that shaped their ornamental structures at a rate that strikes modern researchers as remarkably fast. Triceratops, the last and in some ways most conservative member of its subfamily, appeared around 68 million years ago and retained 3 prominent horns while significantly reducing the elaborate frill ornamentation that characterized its relatives. The spikes, hooks, and projections adorning the frills of its cousins were absent in Triceratops, replaced by a solid bony sheet that was structurally simpler but apparently no less effective for its biological purpose. The horns of Triceratops have been assumed since its discovery to be defensive weapons deployed primarily against Tyrannosaurus rex. The two animals coexisted in the Hell Creek ecosystem during the final 2 million years of the Cretaceous, and direct physical evidence of their interactions survives in the fossil record. T-Rex bite marks have been identified on Triceratops bone from multiple specimens, and the pattern of those marks has been analyzed in detail. Some show no healing. The Triceratops died from or shortly after the attack. Others show bone remodeling around the wound margins, indicating the Triceratops survived the initial attack long enough for healing to begin. A specimen studied by Denver Fowler and colleagues showed T-Rex bite marks concentrated on the back of the frill and the base of the horns, in a pattern consistent with the predator gripping the head and attempting to pull it back to gain access to the neck and throat. The interaction between Triceratops and T-Rex was real, repeated, and sometimes survived by both parties. What the fossil record does not support is the idea that defense against T-Rex was the primary evolutionary driver of the horns themselves. A 2009 study by Andrew Fark and colleagues examined Triceratops skulls specifically for pathologies consistent with horn-to-horn combat between individuals of the same species. The methodology compared lesion rates and lesion positions on squamosal bones, the bones forming the sides of the frill, between Triceratops and Centrosaurus, a ceratopsid with a very different horn geometry that would produce different contact patterns during intraspecific fighting. Triceratops squamosals showed significantly higher rates of healed lesions than Centrosaurus squamosals, and the positions of those lesions matched the contact geometry expected when two Triceratops lock their brow horns together, the horn tips pressing against the frill of the opponent during a shoving match. The wounds were healed, meaning the animals survived the encounters. The pattern across multiple specimens pointed to regular intraspecific combat, most likely males competing for mates or territory, as the selective force that maintained the brow horns at their observed length and curvature. The horns evolved primarily to fight each other. Defense against T-Rex was a secondary benefit rather than the original driver, in the same way that deer antlers evolved for male competition and only incidentally function as defensive weapons against predators. The frill tells a different, and in some ways, more surprising story. Early proposals attributed the large, flat surface to thermoregulation. The frill could radiate excess metabolic heat or absorb solar warmth, functioning like the sail of the Permian synapsid Dimetrodon has been modeled as doing. Detailed anatomical work undermined the thermoregulation hypothesis fairly quickly. The vascular channels preserved in Triceratops frill bone run in a pattern that does not match a structure optimized for heat exchange. Instead, the channel geometry is consistent with blood being pumped through a skin surface for display purposes, producing visible color changes or flushing in the skin covering the frill, similar to the function of dewlaps and throat fans in living lizards. The epiossifications along the frill margin showed a pattern consistent with display structures used for individual or species recognition, rather than structures shaped purely by mechanical function. In living animals, display structures used for mate choice and individual recognition tend to be highly variable between individuals and consistent within individuals, because the function requires reliable signals that distinguish one animal from another. The Triceratops frill fits that pattern precisely. It was almost certainly a visual signaling organ used in species recognition, mate assessment, and possibly individual identification, rather than a thermal radiator or a defensive barrier. The diet of Triceratops has been substantially revised from the early image of vast herds grazing across open Cretaceous plains. The revision starts with the vegetation. Grasses existed in the late Cretaceous, but had not yet spread into the dominant ground cover ecosystems they occupy today. The Hell Creek environment was a lowland forest and coastal floodplain system dominated by flowering plants, ferns, cycads, and conifers rather than grass. The open savanna landscape, so often depicted in Triceratops reconstructions, did not exist. A 2013 analysis by Jordan Mallon and Jason Anderson compared skull ecomorphology across the large herbivorous dinosaurs of the late Cretaceous, mapping feeding niche based on skull geometry, dental battery structure, and jaw mechanics. Triceratops emerged from this analysis as a low browser, an animal feeding on vegetation at or near ground level, selecting for tough, fibrous plant material and processing it with batteries of tightly packed, continuously replaced teeth that wore in a pattern inconsistent with fine grass, but well suited to fibrous stems, palm fronds, and low, woody vegetation. Computational modeling of stress distribution in the Triceratops skull during biting showed a system optimized for lateral slicing of resistant material rather than the grinding motion characteristic of grazers. Triceratops was built for the forest, not the plain. The social life of Triceratops remains one of the more genuinely uncertain aspects of its biology, and the uncertainty itself is informative. Many ceratopsid relatives are known from large bone beds. deposits where dozens or hundreds of individuals died and were buried together, interpreted as evidence of herding behavior, possibly mass drownings during river crossings by migrating groups. Centrosaurus bone beds in Alberta contain thousands of individuals. Pachyrhinosaurus bone beds have been found at multiple sites across Western Canada. Styracosaurus and Ionosaurus are similarly known from multi-individual deposits. Triceratops, despite being one of the most abundantly sampled dinosaurs in the fossil record, is almost always found as isolated individuals. Large Triceratops bone beds are rare and, when found, tend to be small, containing only a few individuals rather than the thousands seen in other ceratopsids. This pattern is consistent across an enormous sample from the Hell Creek Formation, representing millions of years of deposition, and it is difficult to attribute entirely to preservation bias given how well the formation preserves other large animals. The evidence suggests Triceratops was less gregarious than its relatives, possibly living in small groups or largely alone, coming together seasonally for breeding, but not maintaining the large migratory herds that Centrosaurus apparently did. A heavily armed animal whose horns show evidence of individual combat, living in a forested environment where large group cohesion provides fewer anti-predator advantages than in open country, may have had genuinely different social needs from the plain-dwelling ceratopsids further north. Pull all of this together, and the animal that emerges from the past 3 decades of research is almost unrecognizable from the textbook version. Its skull changed so dramatically across its lifetime that the final growth stage was classified as a different genus for over a century. Its horns were shaped primarily by fighting between individuals of its own species, rather than by evolutionary pressure from T-Rex predation. Its frill was a visual display organ that shifted in color and pattern across an animal's lifetime, not a defensive shield or a heat exchanger. It fed as a low browser in a forested floodplain ecosystem, processing tough, fibrous vegetation with continuously replaced dental batteries rather than grazing on open grassland that did not yet exist. It may have been largely solitary, structurally different from the large herding relatives it is usually depicted alongside, and it was present in the Hell Creek ecosystem right up to the moment the Chicxulub impact ended the Cretaceous, its bones sitting within meters of the geological boundary that separates the age of dinosaurs from everything that came after. The DNA that would have settled every remaining question, the physiology, the developmental genetics of the frill transformation, the full phylogenetic position within Ceratopsidae at the sequence level, is gone, and it is gone permanently. The proteins that may survive inside the dense cortical bone of ceratopsid specimens are still being characterized, and whether original molecular material from Triceratops is genuinely recoverable has not been definitively answered. What the fossil record has delivered instead is a century and a half of bone, skull, tooth, skin, and histological data that keeps yielding new answers as analytical methods improve. A species whose adult form was misidentified as a separate genus. A frill that never stopped changing. Horns that recorded the outcome of fights between individuals rather than the evolutionary pressure of an apex predator. A diet built around plants in a landscape that no longer exists. The most recognizable dinosaur silhouette in the history of paleontology turns out to have been concealing a biology that the 3 horns alone never suggested. The closer the bones are examined, the more specific and surprising the animal becomes, and the further it moves from the version on the lunchbox. Subscribe. New discoveries about lost species posted every few days.